Okay, students. Good morning, everyone. Is the screen visible to all? Okay. So, in our last class, we have discussed till vectors. Okay, different vectors we have studied. We have studied about restriction enzyme. Then we are done with vectors. Now, in today's class, I will be discussing about host cell. Okay. See, once we have constructed our desired vectors with the foreign gene, now this vector need to be transferred to a host cell so that inside the host cell, the vector will replicate. Means it will reproduce, it will make many copies of the foreign gene, of the desired gene that we want. So, host cells, now there are different types of host cell. For example, we have E. coli, we have yeast, then plant and animal cells are also there for this purpose, for the purpose of recombinant DNA, or you can say for gene cloning. Now these cells, the host cells, we cannot randomly select any cell and we can put our foreign DNA in that. We need depending on our experiment that we are performing, depending on that, we need to choose our host cell. Now, the most common that we widely used in recombinant DNA technology is the E. coli. So, E. coli is very much used for recombinant DNA technology as a host cell. Now, why we are using E. coli in most of our experiment? Because the first reason is it is very easy to grow and handle. So we can easily grow the E. coli cells in a lab, and uh, students, this screen is not visible or what? Okay, wait. Now it's visible. No, why it's now? Is it okay? Okay. So we were discussing about host cells. Okay. So uh, E. coli, it is widely used in most of the experiment for DNA, recombinant DNA technology. So why we are using E. coli? Because the first reason is it is very easy to grow and handle. So we can easily grow the E. coli cells inside a lab. Second point, which is very important, is that it doubles its cell number in every 20 minutes, means that it can reproduce after every 20 minutes and it can double its amount. Okay, and thus making us possible to clone large amount of foreign DNA. So ultimately we can clone a large amount of foreign DNA with this E. coli, because after every 20 minutes, suppose if I have suppose if I have uh, one E. coli cell. After doubling, after reproducing, it will make it will make 2 E. coli cells. And again, these 2 E. coli cells will divide to make 4 E. coli cells. Okay. So after every 20 minutes, it will keep on dividing, it will keep on multiplying. The next reason is it can accept a wide range of vectors. So whatever vectors we have studied most of the vectors like plasmid vector, plasmid vectors, all these factors, if the host cell, it can easily accept. See, the host cell also, it accept and it reject also some vectors, okay, which it will not allow it to enter inside the host cell. But E. coli, it accepts most of the vectors. Okay, now if it accept most of the vectors, so that so now we can have a variety of whatever vectors we have used, we can easily clone it. We can go for the next step. We can easily clone it with the E. coli. Then the last point, its genetic makeup has been intensively studied. So E. coli, we have studied every in detail. We have discussed its genes and all in detail. So if there's a need, like we need we need to put this gene whether it will accept or reject. We already have studied E. coli in so much detail that we know that okay, this gene, this foreign factor will will be accepted, will be able to work inside the genome of the E. coli, because we have studied all the genes, all the genetic makeup of the E. coli. Okay, so these four reasons why we are using E. coli for recombinant DNA technology. These 4 points you have to remember. In exams, it has been asked uh, why we are using E. coli for recombinant DNA technology. So you have to mention these 4 points. I will be giving you the PDF. You have to write it in your notebook. Then E. coli, we know that it is a bacteria. So it is prokaryotic or eukaryotic? Yes, class. Prokaryotic. Okay. So E. coli, it is a prokaryotic. Now, if we have to clone a eukaryotic protein, if you want to synthesize something eukaryotic protein, we need a eukaryotic cell. Okay, we cannot use a prokaryotic cell that time for expressing a prokaryotic protein. For you can say for making a prokaryotic protein, we need a prokaryotic cells. So for the expression of prokaryotic proteins, prokaryotic cells are preferred. There are various reasons. The first one is that in your last year syllabus, we have studied about the replication, translation, and transcription process. So, in translation process, that is during protein synthesis, the protein that has been synthesized, it needs a proper folding of the protein, and proteins are synthesized from, yes, central dogma. Did you remember? No. Okay, amino acids are the basic block of the this proteins, and for that again we need the from DNA, RNA will be synthesized, and from RNA the proteins will be synthesized. Now, this RNA conversion of RNA to protein is called the translation process, and in this process, when we are synthesizing the protein, we need its proper folding of the protein. We have created the raw protein. Okay, now the folding, primary, secondary, tertiary, and quaternary structure needs to be there. Okay, so proper folding of proteins is taking place in eukaryotic cell. Also, the post transcriptional modification. After translation is over, there are certain modifications. For example, splicing, for example, capping, tailing. Okay, these uh, needs to be done in eukaryotic cell, but prokaryotic cell lacks all this post transcriptional modification. Okay, so prokaryotic cells, if we take the E. coli, E. coli will not undergo protein folding, it will not undergo post transcriptional modification. As a result, the protein that we want, the eukaryotic protein that we want to clone, will not be proper. properly done. So we will be taking eukaryotic cells for eukaryotic proteins, for expression of eukaryotic proteins. The second reason is that your eukaryotic, the host cell, it contains splicing enzymes. Okay, this is again a post transcriptional modification. So the enzymes that are needed for splicing is present in eukaryotic cell, but it is not present in prokaryotic cell, because in prokaryotic cell protein synthesis takes place directly. They don't have to do the post transcriptional modification. Their synthesis of protein is direct, but in eukaryotes we have post transcriptional modification. So what is splicing? Yes, class, what is splicing? Anyone? Okay. So splicing is removal of introns. Okay. In protein synthesis, what we used to do, suppose we got a long segment of protein. Okay. Now this segment contain exon and intron both. And here in the process of splicing, we used to remove the introns, because introns are the non-coding sequences. They will not code for any protein. So we don't want that. The exons, it code for a particular protein. Suppose you take any example of protein, the exon will be coding for that protein. Suppose a protein for insulin, the exons will be coding for that. A protein for growth, the exons will be coding for that. But introns are non-functional. They does not code for any protein. So we don't want the introns. And this exists only in eukaryotes. So in prokaryotes, there is no exon and intron. They directly have the exons, the required protein. But in eukaryotes, we have exon and introns. So the process of splicing, it takes place in this eukaryotes. So here, the enzymes that are required for splicing is present in eukaryotic host cell. So we will be using that, the eukaryotic host cell will be using those enzymes to remove the introns from it after synthesizing the protein. Now, if I talk about the example of eukaryotic uh, host cell that we are using for expression of such gene cloning or expression of protein, is the yeast cell. So, yeast cells, it has been used for the expression of eukaryotic genes. Okay. Now, what are the features why we are using yeast cells? Same that of what we have studied for E. coli, why we are using E. coli. Here also, for yeast cell also, there are some basic characters which you need to remember. The first point is easy to grow and manipulate. So yeast also, it is very easy to grow inside a lab, and we can easily manipulate its gene. Whatever genes we want, we can put those gene. Whatever we don't want, we can remove also. So accordingly, we can manipulate the gene of this E. coli cell. Sorry, of this yeast cell. See, for E. coli also, we will be having some manipulation. Directly, we cannot take the E. coli, okay, for this, any genetic experiment. Certain modification has to be there, which is done by scientists before performing the experiment. Now, second point is well characterized. Again, the genes and the genome of the yeast cell, we have already studied. We know everything about it, the genes that are present in the yeast cell. So whatever we want to grow, we can easily know that this will work in this yeast cell, and we can easily select the yeast cell. The last point is large amount of cloned genes or recombinant proteins can be obtained from the yeast culture grown in a fermenters. So again, the amount of the clone genes. Again, yeast cell will reproduce very fast, and for yeast cell, we are growing it in large fermenters. What are fermenters? These are large vessels where we used to culture the genes. Okay, we will be culturing it. See, in lab, whatever experiment we are doing, it is in small amount. But if you want to grow something in large amount for the whole, uh, you can say, country, or for a huge number of population, we use fermenters. That is, these are very large vessels, so it contains all the requirement, that all the nutrients and all that a cell required to grow. And then uh, there are systems we have connected. So the process used to take place, and we can culture, we can prepare our cells in them. Those, not only for yeast cell, for E. coli also we use the fermenters if we are growing it in a large amount. But for lab purpose, we will be growing it in a small amount. Suppose 100 or 200 cells. Next, once we have got the host cell, uh, see, we have got the vectors, we have got the host cells. Now, what is the main purpose was constructing a recombinant DNA. So, the basic steps, this I have also discussed in our classroom also, in the very first class. The basic steps that are involved in construction of a recombinant DNA molecule is, in the first point, isolation of vector and the fragment containing the gene to be cloned. So the first step is to isolate the vector. We will take out the vector, okay, the whatever vector we want for our experiment. Accordingly, we will take out the vector and the fragment containing our gene to be cloned, and the your foreign DNA also that we want to be cloned, that we want to get expressed. So that gene also we will isolate. Okay. In the next step, the vector and the target DNA fragment are separately digested, that means separately cut with the same restriction enzyme, for example, EcoR1, which generates a sticky end. So, in the next step, what we will do, we will take out, we will take the restriction enzyme, and with the help of restriction enzyme, we will make a cut at the in the foreign DNA and in the vector. And then suppose here I am using the EcoR1. This is a restriction enzyme. And after cutting the DNA, the foreign DNA and the vector DNA, I will get sticky ends. Okay. Remember, we have studied about blunt end and sticky end. So it will create a sticky end. Next, the vector is then treated with alkaline phosphatase so that later in the ligation step, the vector does not self-ligate. Now we will treat the vector with, I'm talking here now only the vector. I'm not taking here now the foreign DNA. Only the vector is treated with alkaline phosphatase. The function of alkaline phosphatase is that it will prevent self-ligation. That means the two ends of the vector that we have cut, again there's a chances that it may join together because it is having its complementary there. So there's a chances that they may again self-ligate, the vectors may again join together. So to stop, to prevent that, we will use alkaline phosphatase. The last step is the cut vector and the DNA fragment are mixed in a suitable ratio and then ligated with the enzyme, that is DNA ligase, to yield a recombinant vector containing the insert. Insert here means the foreign DNA. Now we will, with the help of DNA ligase, we will join the two, the foreign DNA and the vector DNA, and ultimately we will create a recombinant vector. I'll show you the image. Is this visible, students? Is this visible? Okay. So this diagram is also there in your textbook. I want you all to draw it. After I'll instruct you after where you have to write. First listen. So here we have the foreign DNA, and this side on the right hand side we have the plasmid vector. Here I have taken a plasmid. The name of the vector is plasmid. Okay, you can simply write vector also. It is understood. Now the region in the foreign DNA, that is the red color one. This is the desired gene that I want to clone. I want this gene to take out from this any organism XYZ, and I want this gene to get expressed. I mean it to get cloned, to make many copies of this gene, so that this gene will produce the ultimate product that I want. Okay. Now this side I have the vector. In vector, the basic characteristics, that is it has the origin of replication ORI, then it has antibiotic resistance gene. Okay, for this antibiotic resistant gene is required in the further process, where we will be checking that whether our foreign DNA has successfully inserted or not. And ORI, it will help in starting the replication process. Once the vector is placed inside the host cell, this ORI will help in replicating in the cell, replicating the vector cell. And on top, we have recognition site for restriction enzyme. And this recognition site is same for both. You can see for foreign DNA also and for the vector DNA also. The recognition site is same. And that is why we can use the same restriction enzyme for both. See, why we are using the same restriction enzyme? Because our foreign DNA needs to be fitted in the vector. So if the sites are same, then only the foreign DNA will bind with the vector DNA. If the sequences are the same, otherwise it will not, because the vector DNA will not find its complementary on the foreign DNA, and it will not allow the foreign DNA to bind with it. So the recognition site is same for the vector and for the foreign DNA. And then we will use a restriction enzyme, the same restriction enzyme, to cut the foreign DNA and to cut the plasmid. Okay. So in the second step, cut DNA fragment and the plasmid vector with restriction enzyme, for example, EcoR1. Now after cutting, I have got a sticky end in the vector. Okay, after cutting with EcoR1, I got a sticky end. This sticky end will help in binding the foreign DNA. Okay, now the vector DNA is now treated with alkaline phosphatase. Only the vector DNA, not the foreign DNA. Okay, the vector DNA is treated with alkaline phosphatase. Why I am doing this? Because this will prevent self ligation, means self joining of the vector. The two ends still they are complementary to each other, so they may join again. So that's why I'm using alkaline phosphatase for the vector, so that they are not joined. They will not, if they join again, then our foreign DNA will have no place to fit in the vector. Okay, so I have to prevent these two ends from joining again. Now in the next step, I'll do ligation by another enzyme called DNA ligase. So this ligation, after ligating it with the after ligation of the foreign DNA and the vector, the foreign DNA will fit into the vector, and I will got a recombinant plasmid or the recombinant vector. So this is the process of making recombinant DNA. Now this vector will be transferred to the host cell. Understood? Yes, class. Okay. Any doubt? You can put forward your doubt. Yes. Any question till now? No. Okay. See, few points I want you all to remember. Questions are there. It has been asked in the past years that why we are using E. coli for recombinant DNA technology. So you have to write those 4 points, the basic characters, why we are using it. Okay. Then it has also been asked why we are using E cell for eukaryotic cells. See, for E. coli will work for prokaryotic cells basically, and yeast will work for your eukaryotic cells. But E. coli also works in some eukaryotic cells also, where the protein folding and all is not required that time only. But uh, if we are synthesizing something eukaryotic protein, then we prefer yeast only. So they may ask for features why we are using the yeast cell. So you have to write those 3 points. Then construction of recombinant DNA with statement and diagram, the diagram which I have shown, or you can also draw the one which is there in your textbook. It is very important. Then one more point, why we are using, why we are preferring eukaryotic cells for expression of eukaryotic proteins. So for there you have to write down about the protein folding, the post-transcriptional modification which is not available in prokaryotic cell, which is only available in eukaryotic cells, the splicing enzymes which is not available in prokaryotic cells and it is present in eukaryotic cells. So these points you have to write. I am sharing the PDF also in the WhatsApp group. Write that entire thing in your classwork notebook. Okay. 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 And if you still have any doubt, do ask. Not like last year, whatever we have done online. I know you haven't studied. So this year it's more crucial. Okay. You are now in 12th standard. You have to be more serious. If any doubt, you ask me. The whatever PDF notes I'm giving, write it down in your classwork notebook. And yes, students, in the PDF, I have mentioned construction of recombinant DNA. Okay, I have wrote those 4 points. The diagram I haven't given. I will send the diagram also. You need to draw the diagram in your notebook. After writing the construction of recombinant DNA, draw the diagram that I will be giving. Okay, okay. Okay, so in our tomorrow's class, we will discuss how this DNA, the vector that we have synthesized, how they are transferred to the host cell. Okay, that we will be discussing. And your attendance will be marked. So I have recorded those who have attended to today's class. Okay, class, thank you. Thank class.